like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. And I'm right where I belong in my happy place. And I'm right where I belong in my happy place.
Hello, thanks for joining us and welcome to the National University of Singapore Yonglu Lin School of Medicine's Healthy Longevity webinar. We have an exciting show tonight. We're going to hear all the latest in research on aging from Vera Gorbunova. And during her talk, you can use the Q&A function to ask questions, and Charmaine will get to as many of those as possible near the end of the hour. In the meantime, however, I've asked Dong Lu, a PhD student in the Center for Healthy Longevity, to tell us about a recent paper on melatonin and ovarian aging.
Thanks for the introduction. As mentioned, my name is Dong Lu, a PhD student of Prof Brian Kennedy's lab. Today, I will be talking about this paper published last year. Melatonin delays ovarian aging in mice by slowing down the exhaustion of ovarian reserve. The process of ovarian aging is closely related to folliculogenesis, as the mainstream view suggests that there is no germline stem cells in the ovaries to continuously replenish the depletion of ovarian follicles, which would be contained in eggs. Thus, the depletion of ovarian follicle is the main driver for ovarian aging in mid-aged women. Early folliculogenesis starts with the activation of primordial follicles, which would be forming the pre-established ovarian follicle reserve, and this would determine the entire reproductive lifespan in women's life. Once these primordial follicles are activated, they will undergo several stages of folliculogenesis and end up with the formation of small entrant follicles. These small entrant follicles would form the dynamic ovarian follicle reserve, which would determine how many follicles are selected for cyclic recruitment by gonadotropin, or they would be eliminated via atresia. Multiple study has been looking at melatonin as a potential therapeutic method to delay ovarian aging in mid-aged women for its multiple biological and physiological roles, including anti-inflammation and anti-oxidation. For example, this study has demonstrated that melatonin injection into mice could potentially improve the litter size and oocyte number in mice. Thus, melatonin has become a main target to delay ovarian aging in extensive studies. However, the exact mechanism behind melatonin's action remained unknown, and whether melatonin has a direct impact on the process of folliculogenesis was still a question then. This study has firstly looking at melatonin's role in vitro. They cultured granulosa cells, which are the main supporting cells in the follicles, and they found out that the granulosa cells express SNAT, the rate-limiting enzyme in melatonin synthesis, and they can synthesize melatonin on their own. By looking at the figure at the right-hand side, we could see that the levels of melatonin as well as SNAT decrease in an age-dependent manner. They also cultured ovaries from mice with melatonin, and there was a significant decrease in activated dendritic follicles as well. Thus, this part has shown that melatonin regulates early folliculogenesis in vitro. This group of scientists has then moved to in vivo models by injecting melatonin to mice from postnatal day 10. It was also similarly found with a decrease in number of ovulated oocytes, as well as number of small entrant follicles and atretic follicles. At the same time, by mating experiments, there was also a decrease in the implanted embryo numbers, yet with a significantly improved embryo size. In order to investigate and confirm the role of melatonin in ovarian aging and early follicular genesis, they devised the SNAC knockout mice. If you still remember, SNAC is the rate-limiting enzyme in melatonin synthesis. And it was found out that there was a decrease in follicle reserve, as shown by the right-hand side, the top corner, that figure. There was a decrease in the early growing follicles, which indicate there is a decrease in the pre-established ovarian pool. At the same time, there was a decrease in fertility as well, as shown by the figure at the left bottom side. The activated follicle number and atretic follicle number both increased, and this was successfully reversed by melatonin injection, as shown by the decrease in the number of activated and atretic follicles. By mating experiments, SNAC knockout also decreased the fertility of the mice, especially towards the mid-age, as shown by the right-hand side, the corner figure. So this study has shown that the SNAC knockout accelerated ovarian aging, confirming the role of melatonin in early follicular genesis. In a nutshell, melatonin endogenous role in ovaries would include its regulation on early follicular genesis, and by delaying ovarian aging through the inhibition of the primordial follicle activation and early follicle growth and attrition. Potentially, melatonin could be a therapeutic approach to delay ovarian aging in mid-aged women by preserving a better pre-established ovarian reserve. Here comes the end of my presentation. Thank you for your attention.
Thank you. Dong Lu is doing some exciting research in our Asian Center for Reproductive Longevity and Equality, and we'll have someone on to talk about that in the near future. I think it's an important facet of aging that's understudied. But tonight we have Vera Gorbunova, who's the endowed professor of biology and medicine at the University of Rochester in New York and co-director of the Rochester Aging Research Center. She's done a lot of pioneering research in the aging field, particularly in the area of comparative biology, which looks at how different species with very different lifespans age in similar and different ways. She's also worked on SIRT6, which is one of the most exciting longevity genes, and a number of other projects. I could go through publications and prizes, but I think what I'll do instead is turn it over to Vera, because I think we'd all rather hear what she has to say about aging. Thanks for joining us, Vera.
Hello, everyone, and thank you, Brian, for the introduction. So let me begin. I'll share my screen. So I will talk about, some highlights of the comparative biology study today, exactly as Brian mentioned. So first, these are my disclosures, and I'm on the advisory boards of these companies. And with this out of the way, so there are different approaches to study longevity. And the more classical approach is to study short-lived species, and then you can change their lifespan to be longer, but which is convenient. But unfortunately, humans are very long-lived already, so maybe we already have those mechanisms, and we need to look elsewhere. We need to look at species that are naturally long-lived, and they may have mechanisms that are worthwhile adapting to humans. So this is the approach we've taken. And if you even focus on mammals, there is dramatic diversity of lifespans, anywhere from 2-3 years in mice and other small animals such as shrews, to over 200 years in the bowhead whale, and then anything in between. And we can exploit this diversity to understand mechanisms of longevity in the species on this end of the spectrum. So you may be wondering why animals have such a difference in lifespans. So very briefly, I want to talk about evolutionary biology so that everything makes sense in the context of evolution. So here is the force of natural selection. And this is, imagine our lifespan, birth, age at maturity, and then, it could be humans, could be other creatures. The population starts to die out because of accidental death, predation, wars, whatever. So this is so-called selection shadow. Here, the force of natural selection gets weak because there are fewer of these individuals. And this is why genes that would be important to live long, that fall, important at this age, they won't be selected against. So species get to live just as long, to get through this stage. And then whatever happens later is not selected. So for example, for mice, they don't live very long because they get eaten by predators. However, there are many species that have special adaptations that extend this window. And these are the ones we are very interested in studying. So these would be species where accidental death, the probability of accidental death is lower for various reasons. For example, a bat, bats in general live much longer than predicted because they fly, so they can escape predation. Porcupine lives over 20 years, surprising, right? And you can guess why, because who wants to deal with those needles? So it makes its life safer. Naked mole rat, so this is another organism that we study in the lab, is very long-lived because it lives underground. And here there is a turtle, very long-lived because, well, they have a shell. So that's general evolutionary idea. But now how do we use it to benefit human health? So we can either use comparative biology to compare multiple species and look for conserved mechanisms and then see how to apply them to humans. And I will show you examples of this today. Or we can study individual animals, find out what makes them so long-lived. And here we have naked mole rat and whale, and then develop mouse models based on those mechanisms, and from there go to human therapies. So we use that approaches as well. Okay, so now I will tell you about the new project. So this was the work of a very talented bioinformatician, Yu Yang Lu, postdoc in the lab. And what Yu Yang did, he used our collection of tissues from rodents and other animals that differ in their lifespan over tenfold to do comparative transcriptomics. So we sequenced done RNAseq on 5 tissues, actually 6 tissues over 5 individuals of each, and then analyzed transcriptomic signatures of longevity and also identified transcription factors and regulatory networks. So what were the results? So we could identify species which expression negatively correlates to maximum lifespan. So these genes, well, we need those genes, but they shorten our lifespan. And there are 2 major clusters here. So these are metabolic genes. You can see mitochondria, cytochrome C complex, fatty acid catabolism. So everything that perhaps determines the rate of living. And then on the right side, another very strong cluster are immunity genes, particularly cytosolic DNA sensing, innate immune response. So these are, again, these are the genes that would be more highly expressed in shorter-lived organisms. So these are negative genes. So what about positive genes? So that's even more interesting. So here, the most exciting cluster is this one that contains various DNA repair genes, double-strand break repair, homologous recombination. Again, so we can see that across about 30 species, we see that these genes are more highly expressed. So now we have these signatures of positive and negative genes, and we can apply the same signatures to interventions. So here we analyzed data from interventions in mice, so from rapamycin, 17-alpha estradiol, calorie restriction, and we see whether these interventions change genes according to these longevity signatures. And very interestingly, we see, for example, that rapamycin and 17-alpha estradiol actually downregulate negative maximum lifespan genes and upregulate positive lifespan genes. So that's what we expected. So now we could already design interventions based on how they affect gene signatures. Other treatments, you can see, actually don't do what they're expected to do. You know, they still were reported to extend lifespan in mice, but perhaps they provide somewhat a mixed effect. For example, they can upregulate some negative genes, but then upregulate some positive. So maybe they could still work in mice, but perhaps they don't touch the same conserved mechanism of aging. So this may be a great tool for development of interventions. And major conclusion from the study, we also looked at transcriptional networks, and very exciting result was that positive genes are actually regulated by pluripotency transcription factors, which means for longer life, you need to maintain high activity of pluripotency transcription factors, which may be another strategy to achieve rejuvenation. And again, this summary that inflammation, mitochondrial metabolism are negatively associated with lifespan, while DNA repair is positively associated. Okay, so let's talk a little bit more about DNA repair. Why is it so important? Because our DNA is subjected to all kinds of damage, and we have to evolve some ways to deal with it, but this damage can accumulate over lifespan and drive aging. So this is the work of Xiao Tian, very talented student. Now he's a postdoc in David Sinclair's lab. So here Xiao is holding naked mole rat, but I will not talk about his naked mole rat work, but about his double-strand break repair work. So you saw double-strand break repair came as a top hit to be associated with longer maximum lifespan. Because, well, when double-strand breaks are repaired incorrectly, bad things happen. You lose hundreds and thousands of genes, or translocations arise. There are two major pathways to repair double-strand break repair. They're called non-homologous end joining and homologous recombination. So here, Xiao decided to do, not so much bioinformatics work, but actually experimental work to test whether double-strand break repair positively correlates with maximum lifespan using this collection of animals. And indeed, we found very strong positive correlation between efficiency of double-strand break repair. So this was tested in primary fibroblasts from these animals, and maximum lifespan on this scale. So this is homologous recombination, and this is non-homologous end joining. So actually, both of these pathways positively correlate with maximum lifespan. And of course, the question is why? What is the major mechanistic determinant of this correlation? And again, I'm skipping a lot of the details, but let me just show you the conclusion. What we found that this gene SIR2 and 6 was responsible for the major part of this correlation between DNA repair efficiency and maximum lifespan. SIR2 and 6 is a homolog of yeast SIR2. This is a heterochromatin-associated protein, but more generally, it controls chromatin in the cell, and it also, as we showed earlier, is involved in double-strand break repair. It is recruited to the sites of double-strand breaks very early, and then it recruits other factors and is involved in rearranging chromatin at this site. So apparently, SIRT6, just swapping SIRT6 between a short-lived animal and a long-lived animal was sufficient to upregulate double-strand break repair to make it more efficient. So this is the summary of this work where we showed that efficiency of double-strand break repair correlates with maximum lifespan, and we could take SIRT6 from a long-lived animal, beaver, express it in mouse cells, and get beaver-like DNA repair. So that was exciting. So now we actually have a way to improve DNA repair. We know this is a conserved mechanism of longevity, and we can stimulate it. And we know that also it is associated with longer lifespan across species. Now, the question is, what about within species? What about humans? If we increase SIRT6 activity in humans, will it be associated with longer lifespan? And here comes this collaboration with Xiao Tian at Columbia University, and also this is Matt Simon, a postdoc in my lab, who led the study. And we asked the question, is SIRT6 associated with, can we find high activity of SIRT6 in human centenarians? So here I'm showing you some of the centenarians, some of local centenarians. So here is a lady who lived to the age of 119. And this gentleman is actually from New York. So even in New York, people can survive beyond 100. His name is Alex Image, and he stayed active. Here he is at age 111. He was still going to his office and writing books. So this is the kind of healthy longevity we all want, right? So we took a population of centenarians, actually from New York area, and using sequenced them and looked for polymorphisms in multiple genes, but actually she found one very interesting polymorphism in SIRT6 gene. So this is a centenarian SIRT6 allele. There are two missense mutations in the C terminus. We took those mutations, we engineered them, and then tested how they affect DNA repair. And to our excitement, centenarian version of SIRT6 showed more efficient both homologous recombination and non-homologous end joining. So then what's different about this protein? It has two enzymatic activities. So SIRT6 has two enzymatic activities. It can deacetylate protein substrates, and it can mono-ADP ribosylate protein substrates. So interestingly, we found that this second activity was enhanced in the centenarian protein, which was unexpected because this deacetylation receives much more attention. It's been studied for much longer, but actually it was even slightly lower in the centenarian version, but this second activity was enhanced. Another interesting fact about centenarian SIRT6 protein was that it was interacting more strongly with Lamin A. So Lamin A is a very exciting protein. It provides the scaffold to the nucleus. And the mutation in Lamin A is associated with multiple genetic diseases, but the most well-known one is Hutchinson-Gilford progeria, because it's a very severe childhood progeria. These children die in their teenage years, and in these children, Lamin A is malfunctioning. However, there are also polymorphisms in Lamin A that are associated with centenarians, so you can actually even have a better version of Lamin A, you can have a worse version. And we see that centenarian SIRT26 binds more strongly to Lamin A. So here, our model for that, that somehow when centenarian SIRT6 binds to Lamin A, it helps support better maintenance of the nuclear architecture. And there is less epigenetic drift, so-called, with age. So what is epigenetic drift or epigenomic drift? So that must be something very sad. When the cell is young, chromatin is packaged very nicely. So some areas are tightly packaged into heterochromatin, other areas are open, and the epigenetic landscape looks very sharp, like there are hills and valleys. But as we get older, because of repeated cycles of DNA replication, repair, heterochromatin starts to unravel. So it's almost like you have your underwear drawer and you fold it very neatly, in the beginning of the week, but then as you keep going in and out, things get rearranged. And so that's pretty much what happens to our epigenome because of DNA damage, transcription, and everything. So epigenetic landscape gets smoothened. And this is perhaps why we see genes associated with double-strand break repair come up as being important for longevity, because double-strand breaks would also lead to this loss of sharp epigenetic landscape. So now the question is, okay, how do we go from here to interventions? Because it looks like, yes, SIRT26 function is associated with longer lifespan across species and within species in human centenarians. So as a first proof of principle, we tried to overexpress SIRT6 in fibroblasts from human individuals of older age, around, starting age 60 to 70. And then after 2 weeks of SIRT6 overexpression, we measured methylation age using a methylation clock developed by Steve Horvath. So here you can see HPRT was just our control gene that we expressed. So when we express, so this is expressing HPRT and the same cells from the same individual expressing SIRT6. So after 2 weeks, DNA methylation age went down. So that was really, really exciting. We've also done RNA sequencing on the same samples. And these are transcripts that were upregulated following SIRT6 overexpression. And you see most of the top categories are DNA conformation change, chromatin assembly, DNA packaging. So indeed, we see that expressing SIRT6 stimulated all of these processes and perhaps repackaged chromatin into more youthful conformation. So this was a genetic approach, but with humans, we want to use pharmacological approaches. And then one way would be to develop activators for SIRT6. And people have been working on it, but mostly focusing on activators of deacetylation, just because that's been the activity most characterized. So here we collected activators that's been published in the literature reported to activate SIRT6 deacetylase activity. And then we tested whether any of them also stimulate mono-ADP ribosylation activity. This is the activity that's enhanced in human centenarians. And you can see that most of them, so this is the controls, most of them actually don't. So you need to look for separate molecules if you want to activate one activity or the other. But one molecule activated both. And you may be wondering, what is this? So this is a very interesting molecule. It may be not very exciting from the drug design point of view because it's not a small molecule, but it's a natural molecule. It's something that can be taken as a supplement. And it comes from seaweed. So this is called Fucoidan. It's a short polysaccharide found in seaweed. And people already consume a lot of seaweed, and there are different health benefits associated with it. And very remarkably, there are Asian countries with very long life expectancy, would be like South Korea and Japan, where consumption of seaweed is very high. So we decided next to test whether it can actually slow down aging. And I'm showing you a very fresh result from our mouse studies. So here we took mice, aged mice at about 20 months of age, and we gave them Fucoidan for 2 months, and then we tested their frailty scores. And you can see control mice versus mice given Fucoidan. So these are males, and these are females, female controls, females given Fucoidan, and these are both sexes combined. So you can see after even 2 months, we see reduction of frailty scores in the mice given this supplement. So that's very promising. I can recommend seaweed to anyone. At least what I can tell you, it won't do any harm. So let me conclude with this scheme. So what emerges from molecular studies and comparative omics studies, that chromatin organization and DNA repair are very important for longevity. Our epigenetic landscape unravels as we get old, and now we need to find ways to put it back together to achieve actual rejuvenation. And there are different strategies here to achieve epigenetic rejuvenation. So one strategy that's been discussed quite broadly, and there were exciting results reported by several laboratories, was using partial reprogramming using Yamanaka factors. So this is a very powerful method, but there is a danger because cells may lose identity and give rise to cancer. So interventions using partial reprogramming will need to be very safe before they can be applied to people. And here we are offering an alternative to repackage epigenetic landscape, but without changing cell identity. And this can be achieved by activating SIR2 and 6, or perhaps by targeting other epigenetic enzymes that will just refresh the chromatin structure, but not completely rewire it as it happens in embryogenesis. So I guess I'm 3 minutes ahead of time. So, okay, will also give acknowledgement to people who contributed to all of this work. I was naming people as I was going along. This is my lab at Cold Spring Harbor. I showed work by Xiao, Matt. Yu Yang is not here yet because he joined later. All of this work was done in collaboration with Andrei Siluanov. And here also are our external collaborators. We are mostly funded by the NIH. We have program projects on comparative biology of aging, a program project on heterochromatin and the role of line 1 elements that are actually also silenced by SIR2 and 6. So when I was talking about packaging heterochromatin together, important part of it is actually packaging line 1s back into heterochromatin so they don't get activated. And then some other exciting collaborations. So this is U19 with Jan Weig and Yusin Su, the Centenarian study. And now we are also participating in another Centenarian study with Paolo Sebastiani and Tom Perls. Thank you very much. I'm happy to take questions.
Thanks, Vera. That's a great talk. I think that that brings up a lot of questions and where to start. So I think maybe let's start a little bit general. You've thought a lot about comparative biology of aging, and there's been a lot of debate recently about whether maximum lifespan in humans can be affected. I think all of us believe that interventions are likely to affect median life expectancy and health span, but what about the maximum? Is 120 the end result here, or do we need to find new mechanisms like whales have, and how do we get past that?
Yeah, that's a very important question. Well, 120 has been around since biblical times because it also can be found in Genesis. So it has been the limit. And I would think that when you're asking whether interventions can move this, it really depends on what interventions. If we think about interventions that are more of supportive nature, like antibiotics, better hygiene, maybe even some dietary changes. So they will probably affect medium lifespan more, and we'll have hard time moving end threshold. But if we target the mechanisms of aging themselves, my belief is that we can actually make people live even longer. But probably until we get to that point, we'll just be happy to increase mean lifespan. If everyone lived to 120, wouldn't that be nice? But I think if we actually targeting the mechanism of aging itself, or if we can achieve actual rejuvenation, so then I think 120 will change to a different figure.
Yeah, I mean, it may be possible that unless you get close to that by dealing with other problems, dealing with the problems at 120 may not matter that much. So it's unclear, I think, whether reprogramming would fix everything or only things that come into play later. So it's an interesting question. Another thing I get asked all the time is, because this has been debated in the literature, is do naked mole rats age or not? What do you believe? Because there was a paper published that shows no signs of aging demographically for naked mole rats. What's your thoughts on that?
Oh, yeah, thank you for that question, because we just published a paper, actually 2 papers. So we published 2 papers, one in Nature Aging, one in Nature Communications with Vadim Gladyshev, and Nature Aging with Steve Horvath, where we addressed exactly this question using a methylation age. And we both studies using different methodology for methylation age arrived to the same conclusion, that epigenetic age increases with age in naked mole rats. So epigenetically they age, but it is somehow uncoupled from physiological aging, or at least physiological aging is very much slowed down. So maybe this is yet something else we can learn from naked morat, how to uncouple these two processes.
It reminds me a little bit of the review we wrote in bats, right? Where they have bats live a long time, as you mentioned, and it may be because they still have signals for inflammation, but their response to inflammation is uncoupled from that a little bit. So I don't know, you want to comment a little bit about bat aging?
Oh yeah, I'm fascinated with bat aging. We now have multiple bat cells in the lab, and we are also collaborating with Linfa on that. So yes, one, I think, very exciting result about bat aging, which is something we highlighted in the review that we wrote 2 years ago, was that they are missing this whole set of genes responsible for DNA innate immune sensing. And here from our comparative omics study, we see, and we analyze species that are not missing this group of genes. But this group of genes came up as being one of the strongest negative associations with maximum lifespan. So you actually need to get rid of those innate immune sensing genes. And of course, the question is, how do you do it and at the same time stay resistant to infections? But bats obviously know how to do that. So this is another very exciting model for aging research.
Well, as I understand it, the bats often, they're not necessarily resistant to the infection. It's just more of a chronic thing with them that I don't know if that's true or not. That's what I've been told about it, that they still carry viruses and they're good at spreading them, but they don't, it doesn't cause them to die from it. So.
Yeah, so they can somehow develop tolerance. If a human strategy is to develop very aggressive inflammatory response, but again, maybe we are just not as good as bats at dealing with them, because in bats, we've done additional experiments and we see that if you challenge bat cells with some inflammatory stimuli, they either don't respond or they can respond very quickly, very robustly, on much faster kinetics than human cells. So they probably just get rid of it. And in human cells, there is a bit of a delay and then very robust response, so, which may be not ideal.
Interesting. One more comment on comparative biology before we get into more detail on sirtuins and other things. You looked at mechanisms of long lifespan in a lot of different species, and you alluded to some of them. I think that you would conclude that each of these long-lived species have developed some of their own unique mechanisms for getting to long lifespan. It's not like everybody converged evolutionarily on one pathway. So what does that mean? Does that mean there are a lot of different ways to live a long time, and that many of these things will work if we could translate them to humans, or that every species has to develop its own mechanisms, and maybe these things won't work so well if we try to translate them? I know there's limited data on that, but can you speculate?
Yeah, that's a good question. So the picture that emerges, because in the beginning of my career, when we just started looking at species, we thought everyone should choose the same path. And this is often a misconception that people working on model organisms, because they only look at things that are conserved. So it seems like there is only one strategy. But by looking at the diverse long-lived species, we see that there are also unique strategies. And I think what confers longevity to any given long-lived organism, be it a whale or naked morat, is a combination of more unique species-specific mechanisms and conserved mechanisms. And there's some examples. We talked about DNA repair today. We talked about suppression of inflammation. So these are conserved mechanisms. But there may be, first, how do you arrive to that? So bats, for example, just throw away this bunch of genes, right? So this is a bat-specific thing. Naked morat may upregulate hyaluronic acid, which actually has anti-inflammatory properties in itself. So there are all these different strategies, and often the end result may be the same, like in this example, reduction of inflammation, but the way they get the species evolve it is different. So this is why I think it's very important to study different species, and the mechanism may be different, or the means to achieve the results will be different, and some of them may be easier to translate to human than others.
We have a lot of people that are taking different supplements and interventions, and you mentioned hyaluronic acid. So give me a short version of why hyaluronic acid is good for naked mole rats and whether it makes sense to go buy it at the supplement store.
Well, hyaluronic acid, so I could give you a whole other seminar. We have really exciting results on hyaluronic acid. Naked mole rats have very high levels of hyaluronic acid in pretty much all the tissues, and that's from connective tissues, blood vessels, heart, brain, everywhere. There is 10 times more hyaluronic acid than in human tissues. And also the molecules are much longer, which gives hyaluronic acid different properties. It's more viscous. It has different effects on cells. And what are these beneficial effects? So it's anti-inflammatory. It's anti-metastatic in the case of cancer. And we also recently found that it can preserve stem cells better. We engineered transgenic mice that express naked mole rat hyaluronan synthase gene. And these mice have reduced inflammation, they live longer, and they maintain much better intestinal health. And it took us a while to figure out why the intestines are better maintained, there is less leaky gut in old age. And then we found that actually the intestinal stem cells are kept in much better shape by hyaluronic acid. So there are all these different effects. The question, can it be achieved just by oral supplementation? I honestly don't know. There are some studies in the literature that say that oral supplementation can be beneficial. But ideally, what we would want to do is upregulate hyaluronic acid internally, because it's a very large molecule and it's better to slow down degradation. And that's something we are developing right now.
Oh, that's exciting. I was just a couple questions on the positively regulated genes and the negatively regulated genes between species. It seemed like a lot of them, like rapamycin, behaved as you would have expected. Some of them behave oppositely to what you would have expected. The one that jumped out at me, though, was calorie restriction, which seemed to positively affect the positive genes or upregulate the positive genes but do the wrong thing to the negative genes. Have you thought about what that means for calorie restriction?
Yes, perhaps it means that it has benefits, but it also has some disadvantages. So there are problems. And that may be kind of a mixed bag, that calorie restriction may induce certain stresses, certain metabolic stresses that are not necessarily good if we are looking cross species. Maybe for the mouse that is calorie restricted, generally the some result would be beneficial, but if we want to design an intervention that goes along, the strategies that evolve to provide much longer lifespan to long-lived species, so you perhaps need to stay away of calorie restriction or maybe modify it in a certain way. Because the longest-lived species we know are not particularly calorie restricted, like think about the whale. So I think there are certain interventions here that they're a mixed bag. And again, maybe I will say something controversial, but the mouse is probably a very good model to see benefits of calorie restriction, because this is an animal with very fast pace of life. And here, calorie restriction effects would be the most pronounced, because you just slow it down a little bit, then will live longer. But for an organism like human that already has pretty slow pace, there will be some benefit, but perhaps not as pronounced as for the mouse. So again, that would be a motivation to look for something different.
You also had circadian rhythm genes on there and associated with the negative genes. And so is there a story there? What does that mean?
Oh, yes. Well, I compressed the story. What we found was that negative genes, when we look at transcriptional networks, they are controlled by circadian transcription factors, which means that those metabolic genes, especially in long-lived species, their expression is restricted, perhaps, to a certain time of day. And that may be the way to down-regulate them, to limit the expression of those genes for longer lifespan. And the implications for humans would be that, yes, we need to respect our circadian clock. And maybe the study that was presented by a student in the beginning, melatonin would be one of the factors to kind of restore the balance of circadian clock somewhat and perhaps compensate for unnecessary light exposure. So I think that directly connects to it because it may be down-regulating those negative lifespan genes.
Well, Singapore will be in good shape then because the light never changes, the time never changes. We should be good at circadian rhythms here. And actually, I guess this might be the first act of Congress that directly targets aging then, because the U.S. government is thinking about getting rid of the time shift with daylight savings and just going to
I'm so
permanent
excited.
daylight savings.
I hope they approve it. But, of course, daylight saving was a disaster. It's not good for aging. But I think human behavior is also very important, what we do if people get, exposed to bright lights at night. So that also would disturb your circadian rhythm, upregulate your negative lifespan genes.
I got a question I have to ask because a lot of people that listen to the show takes NAD precursors. So, and NAD is involved in SIRT6. So, how much? We've talked about this, but just here for everybody else's sake, how much is an NAD precursor likely to activate SIRT6, especially with age?
Well, among all the sirtuins, there are 7 of them. So SIRT6 is one of the least sensitive to NAD. So I would say, yes, it is a cofactor. It's important to have NAD, but whether those minor fluctuations in cell NAD that we are talking about in aging are particularly important for sirtuin-6, I'm not convinced. And this is why we are looking for other strategies to activate.
Great. I think I could ask questions all day, but maybe I'll bring Charmaine in, who's become a regular now on the show. This is like 4 or 5 times you've been on, right? I mean, she's a total professional. She's going to leave my lab and become a news anchor at a major TV network or something.
Dual career.
Okay.
So what are the cool questions tonight, Charmaine?
Yeah. Hi, Prof. Thank you for the talk. We have quite a diverse set of questions, so I'll just ask one of the more popular ones that was voted. So this is from Vincenzo, and he asks, what could be your opinion? What's your opinion on the reason why mitochondrial and metabolic pathways would be linked to a shorter lifespan? And do most of the shorter-lived species have higher mitochondrial activity or metabolic rates?
Yeah, so I would say a simple question. We could say, well, in general, there is this very old and debunked idea of rate of living theory that if you have higher metabolism, so you live faster. And to a certain extent, what we see could be a supportive of that. But of course, there are many exceptions. And we talked about bats. When bats fly, their metabolism gets really, really high. But then they have some strategies to counteract it. So we see, again, because aging is such a complex process and longevity is composed of, combination of different mechanisms. So we can see, yes, slowing metabolism perhaps is good, but then you have to also take care of inflammation and DNA repair. So I wouldn't make such blanket statements as, well, all short-lived species have high metabolism. No, not necessarily. Just one of the components.
Okay, thank you. So I think a lot of people are also very interested in the seaweed and Fucoidan. So I think these 2 questions are quite linked together. So the first question from Matthias asks you if how is the uptake of this compound in seaweed facilitated on a cellular level? Since you mentioned it's not a very small molecule, right? So how is it taken up in the cell? And do you plan to work and look at the mechanisms of Fucoidan in the future?
Well, this is a very important question, and we were concerned about it as well, because when we first started working with Fucoidan and we saw very strong in vitro activation, and then we thought, oh, will it even get inside the cells? And we've done some experiments, and we see that it does penetrate the cell. We don't know exactly what the mechanism of transport is, but it does get inside the cell, and we even see effects on the whole mouse. but I don't know how. So that's perhaps something needs to be studied and maybe could be optimized. I should also say that Fucoidan is a rather diverse group of saccharides, because there are different species of brown algae, and each one makes a little bit different versions of the saccharide. Not all of them activate sirtuin-6. So we test, and then often, there would be about 30, like 3 out of 10 that do it. And we are also looking at particular structural features that are responsible for SIRT6 activation, and eventually perhaps we'll know how to optimize it.
You're at a house on the ocean now. Are you starting a seaweed harvesting enterprise, or?
Well, there is a lot of seaweed around. You just need to find the right one.
Okay, thank you. That'll be interesting to find out actually which particular compound on seaweed is responsible. Yeah. So the next one that is quite interesting is someone asked, how strong do you consider the genetic evidence for the SIRT6 mutation? And is the variant that you find only specifically for centenarians, or do you find them in other populations as well?
Oh, this is a very good expert question. Yeah, so many genetic association studies in centenarians are suffering from the same problem, insufficient sample size, because to have very strong genetic evidence, you need more than 10,000 people in your population, and these are very rare alleles. So what I present, it's not to say that all centenarians carry this mutation. It's still a small percentage of centenarians. So we had a difficult time, making a very strong genetic statement, because we see an increase in centenarian versus controls from the same ethnic group, but still the numbers were too low. So to solve this, we went to gene database of like all people, different ages, and we looked at genes that are more represented in older people. So which means, people, let's say above age 70 that survive to that age. And this variant comes up as within the top 5% of SNPs that are overrepresented in people of older age, and not centenarians, but just older age. So that actually reinforces the idea that indeed there is a true genetic association.
It seems like the evidence on centenarians really is that there's not one. It's just kind of similar to the long-lived comparative biology question, that they're not all choosing the same mechanism, that there are a bunch of different mechanisms that are all rare on their own, but centenarians find one of them to get there genetically. Is that how the data, does the data support that?
Or maybe multiple mechanisms, right? There are many combinations of alleles. And yeah, in some people you need a certain set that takes you past 100, and other something else. Yeah, that makes genetic studies very complicated, but I think the field will overcome this difficulty now that sequencing becomes much more available, and they just need to recruit sufficient numbers of centenarians. And once you are past about 10,000 people, so then strong genetic associations can be identified, because up to now, the best one that was found with very high degree of certainty is the protein linked to cardiovascular disease from Nir Barzilai's work, which is of course very exciting because people are resistant to heart disease, but on the other hand, it doesn't tell us about the mechanism of aging. It's just a condition that kills people.
Mm-hmm.
Thanks, Prof. I think I'll move on a little bit. Some people have questions about, you talked a little bit about double-stranded breaks during aging. So this is question David asks, what are some of the major causes of double-stranded DNA breaks during aging in humans? And perhaps a little bit related is also, are there any gender differences with regards to the incidence of getting double-stranded breaks in humans?
Well, there are many causes of double-strand breaks. Of course, there are maybe external causes when people undergo chemotherapy and radiation, but in more sort of normal situation, just from cellular metabolism, DNA replication, acts of transcription. Enzymes can get tangled a little bit in the DNA, and it needs to be broken to untangle it. So it's a product of normal cellular physiology. We cannot completely avoid it, and that's why double-strand break repair is very important. With regard to gender differences, yes, there are differences. In our study, we didn't really focus on it because we had about 5 animals for each time points, and we tried to keep, 2 males to females, so we pulled them together. So I cannot comment on that.
The thinking used to be that maybe homologous recombination was more important for aging than double-strand break repair because it was less mutagenic. But it seems like most of the mechanisms you're talking about, both of those are upregulated. Is there any differential between homologous recombination and double-strand break repair in like a 36 context or other?
Well, so yeah, double-strand break repair has 2 pathways: homologous recombination
and
homologous
end joining. Sorry, yeah.
Homologous recombination is active in dividing cells, and during DNA replication, it's very important. So mutating homologous recombination is almost incompatible with cellular survival for mammalian cells. So it's important, but for cells that are post-mitotic, this is not the pathway to use because you need sister chromatids, so their non-homologous end joining becomes more important. And yeah, you're right. People tended to think, you just shift the balance towards homologous recombination, everything will be good. But, this is more simplistic way. It may be true for yeast that just keep dividing. So there is always this choice available. And of course, they better choose homologous recombination. But for human body with many post-mitotic cells and also very large genomes, because for homologous recombination, you need to recombine with the right spot. But if you recombine with the wrong spot, and there are all these repetitive elements everywhere, so the consequences will be disastrous. So it shifted more in high organisms towards non-homologous end joining, but both of these are still very important. So that's why I cannot say, well, this is good, this is bad. They're both important. And what we see from our comparative study, that both of them are upregulated in long-lived species, and SIRT6 is involved in both.
Is it just, is it the accumulation of mutations that matters, or the disrepair of breaks that's important?
Well, disrepair of breaks will lead to large mutations. So again, people were mostly looking at point mutations, and we know that point mutations accumulate with age. It's easier to score point mutations because with current method of high throughput sequencing, when there are rearrangements caused by misrepair of double-strand break, it's very easy to mistake them for library artifacts. That's why it's a little bit lagging behind in terms of quantifying these events with respect to aging. But all the evidence points to that these events are actually much more consequential because they not only change the sequence, but they also disrupt the epigenetic landscape. And that may have much, far-reaching consequences.
Charmaine, do we have 1 or 2 more questions?
Yeah. Okay. I think this is something that was asked quite frequently. So everyone's really curious, based on all the studies you've been doing on aging and comparing species, are there any supplements or strategies that you have picked up that you personally believe in or you would recommend?
Well, we talked about seaweed, right? So I can recommend seaweed. It definitely tastes good, and we even see the benefits. But by activating SIRT6, which would be something, we understand now the mechanism of it. What else can be done? Based on the research I presented today, I would say, keeping your circadian rhythm in order is very important. Just make sure that at night, you keep your room dark and don't unnecessarily disrupt your circadian clocks. Fighting inflammation is important. So perhaps different compounds that can target inflammation have great potential. But again, it's not something I can recommend right away because many current anti-inflammatory drugs have other side effects. But perhaps any compound that is safe and may have anti-inflammatory properties so that there is some benefit there. Well, with color restriction, we got to this mixed bag effect, but I would say, not overeating is definitely beneficial. So maybe not particularly restricting yourself, but let's say if you skip a meal, so that would be beneficial.
Maybe we better stop there just for the sake of time. Thanks, Charmaine, and
Thank you.
thanks, Vera, for doing this. We'll have to do another show to talk about the projects you didn't get to talk about today. We'll get Andrea on too sometime soon. So I want to remind everyone to use the chat function and choose the panelists and all attendees option to leave comments and feedback on the show, and look for news from our center and our School of Medicine in the end credits. We're going to take a couple weeks off. We'll be back April 7th. The speaker will be Panastopoulos, and the host will be Andrea. And I'm going to leave you tonight with a apropos video comparing the lifespans of various species from ants to elephants. Thank you for joining us.
Like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down.